thanks everybody for coming. As Sandy said, my name is Brant Faircloth. My research group is at LSU. If you have no idea what I'm actually talking about, because my title is a little confusing, I'm talking about assembly errors in phylogenetic imprints. My title is actually sort of a take on the famous palindrome Able was I or I Sell Elba, which involves Napoleon. And I sort of picked it because it was National Palindrome Week earlier this week, and if the timing for my talk had been perfect, it would have been on one of these sort of palindromic days that we had in this year. It was not, but that's just the way the timing works. The reason that I've got this picture up here is it kind of explains how I feel right now at this very moment with respect to assembly error and how it affects phylogenetic or phylogenomic imprints. What I'm going to tell you is this story. It's kind of started off as a toy example that I wanted to play with to see what was going on with assembly error and phylogenomics. So when we think about typical phylogenomic workflows, usually we collect some sort of sequence data. We generally do some sort of assembly or alignment of those data. So I'm going to focus primarily on assembly of data, using things like Trinity or Abyss or Velvet to put those data together in contigs, versus using something like BWA or Bowtie to align those data to a given reference sequence. Okay, so I'm just not going to talk about the alignment type of analysis. After we assemble those data, we generally make alignments from them, and we use those alignments to infer relationships. This isn't anything new for anyone in this room. You've probably all done this or seen this in some way. And what we're basically using all of these data for are to try and infer something about the evolutionary relationships between different species or groups of species. So Jessie did this in her owl talk, Carl did this in the songbird talk, any number of people have done this in their talks at this meeting. And a lot of times when we're talking about inferring evolutionary relationships, the sort of things that we tend to worry most about have to do with generating alignments or inferring some tree from those alignment data. And it's one of the reasons that we typically use or include many different types of analyses in the papers that we publish or the talks that we give. We're trying to deal with this sort of uncertainty that may arise as you use different techniques to analyze the data. And I think as sort of everyone in the room knows, there are many discussions of this uncertainty in the literature. Right? You can dig up many hundreds of papers having to do with alignment error or inference error, and more papers come out every year. Now, one of the things that we don't tend to talk about a whole lot are things like generating our sequence data and putting those sequence data together before we do anything with them. And it's a little bit strange because these steps affect everything downstream, right? So if we're putting garbage in, we're basically getting garbage out. And one of the questions is why is it that we don't, or maybe we don't talk about it quite as much as these other problems that we constantly discuss? And I think part of the reason that we don't talk about these things is mostly historical, right? We're moving into the age of phylogenomics, or you might say that we're there, but we haven't been there for very long. And prior to being able to generate gobs of data and try and figure out how to analyze those, we were basically using PCR and relatively simple assembly methods to put together the data that we generated from PCR-based analyses. Right? So when we're using these simple methods of putting our sequence data together, we're not really worried about them. They're relatively simple. We understand well how they work. The data were also few, and generating consensus contigs was basically an easy thing to do. Now, as we've moved into collecting phylogenomic data, we're getting lots and lots of short sequence read data. We're putting it together with very complex assembly algorithms that I don't know how many people in this room fully understand, but I'm guessing it's a relatively low number. And then we're using these complex assembly algorithms in really intricate processing schemes, right? So sometimes this is just a complex algorithm and we put a little bit of data in it, but sometimes we have so much data we need to divide that up. We're using divide and conquer approaches on really big supercomputers. And again, many people don't know the intricacies of how that works either. And so one of the things that I started to wonder about was whether this assembly process, using complex algorithms, putting in lots of data, doing things that we don't entirely understand, affect these downstream inferential things that we're doing with the data that we collect. So what I'm talking about are what I sort of term assembly errors, which is really a generic term that incorporates both sequencing error, which is about 1% on Illumina platforms, and the process of putting those reads together de novo to form some contigs that inform our analysis. And just to sort of get a sense of if this might even be problematic, one of the first things that I did is I did sort of a simulated assembly problem, right? Using some data from the chicken genome, right? This was Gallus 4, so the most recent assembly of the chicken genome. I started off making the assumption that the chicken genome is perfectly assembled. Now we know that that is not true, but sort of for the purpose of this analysis, it doesn't matter. So I have this baseline genome that I started with. I simulated read data from that genome. I did that using two programs, ART and WGSim. And once I simulated data, I also made several data sets that corresponded to a number of reads on the sort of locus targets that I was shooting for, right? So I just did 20, 30, 40, 50, and 60% reads on target. The off-target reads were randomly drawn from the genome or drawn from the mitochondrial genome. I then took three assemblers that people tend to use, and I don't mean to throw these three assemblers under the bus in any way. All assemblers perform strangely when you see the slides in a minute. I just picked these three. They're the ones that I reassembled the data that I started with, that I'd simulated from the chicken genome, using these assemblers, and I compared these assemblies back to what I started with. I did the comparison by basically aligning my new data that I reassembled from simulated reads to the chicken genome that I started with using math, and I simply computed the number of differences between what I assembled and what I started. Now, sort of a sanity check to make sure that things weren't going completely strangely, I basically established a baseline by not introducing any sequencing error to the reads that I simulated. And when I put those back together, I basically saw that all of the assemblers were performing relatively similarly. So across Abyss, Trinity, and Velvet, and the color scheme will stay the same throughout the rest of the slides, the number of loci that I recovered was roughly the same across all of those assemblers. And the reason that these numbers might differ a little bit is that some assemblers will put a locus together in duplicate, and I was just tossing that out with an error. Okay. Similarly, most of the assemblers put loci together in roughly the same size pieces. Right? So this kind of shows that the assemblers are performing similarly when put in data that have very little error. However, when you start to look at accuracy of the assemblers, right, not just how many loci you're getting, but are those loci what we started with, we immediately see with no sequencing error added to those data to begin with at all, that Trinity, in fact, is introducing a number of differences to the data from what we started with. Right? The other two assemblers perform as you'd expect. Right? There are no simulated error in these data. We should see all of these numbers being very low. Okay? Probably a better way to talk about assembly error is to normalize these numbers by the total assembly length, so we get a relative rate of assembly error. Right? So that's what's going to be on the bottom of these slides. So on the top is just the total number of errors that you get. On the bottom is this relativized rate of error. So once we start adding assembly error, we see some really strange behavior. We start to see the number of loci change as the reads on target increases from a low amount to a high amount. So as we're increasing the amount of reads on target, Abyss is giving us somewhat more loci as we get more data on target. Strangely enough, Velvet's actually performing in the opposite direction. So as we're giving it more data that are what we want, it's actually assembling fewer and fewer loci from those data. Now again, this is sequencing reads that I've simulated where I've added error to those reads. We also see that there's a pretty big difference between how these assemblers are performing in terms of the length of contigs. Right? So Abyss is where we're getting more data that are what we want. This is shorter and shorter contigs. And Trinity, Velvet is just kind of performing at a baseline level that's not very good relative to the others. None of these slides are actually the scary slides. The scary slide is this one. In this slide, it's showing the number of differences that we get when we have simulated sequence reads with error in them, and we put them together with the different assemblers. Trinity, for 20% reads on target, has introduced 13,000 errors to the assembled data that did not exist in what we started with. Once we relativize that, we actually see that Trinity, because it tends to put together a longer loci on average, actually has a slightly lower relativized error rate compared to Velvet. So sort of the worst performer in all of these is Velvet for this case. Now you might be saying, I haven't said anything about sequencing coverage, I haven't adjusted for sequencing coverage, and maybe it's just the fact that all these errors are due to sequencing coverage. So I went back and I trimmed all the contigs such that all of the contigs had greater than 3x coverage ends, and all of the contigs had greater than 5x average coverage. Once I did that, we still see that there are an enormous number of errors in the things that we put together relative to what we started with, and we again see that Velvet remains worse when you look at these sort of relativized error rates. You may also be saying, well, maybe it's that this ART read simulator is simply crazy. Maybe it's just a really bad read simulator and it's doing strange things. So I also used the read simulator WGSim to simulate reads from the genome and add error to those. I put those back together, and basically we see that all of the assemblers are performing strangely again. We're getting a number of differences in the data from what we started with. Here, Abyss, which has been performing reasonably well, starts to perform a little bit worse than it has been, right? And Velvet, if you look at the total number of differences, looks like it's actually performing pretty well. But when you look at the relativized rates, Velvet actually has just put together about 10 contigs that are about 100 base pairs long, and those 10 have a lot of pairs in them. So the relativized rate, again, for Velvet is pretty high, and it's also high for Trinity and relatively high for Abyss. So all of this is pretty scary. And right now I can kind of hear this primal scream, right? Because here's just another thing that you need to worry about. We worry about alignments, we worry about inference of phylogenies, and now we have to worry about this sequencing. But maybe you don't. And the reason that you might not need to is the thing that we're interested in all of this is inferring evolutionary relationships and doing something with those inferences that we generate. And so what I wanted to know is, does this actually make a difference when we're trying to infer phylogeny? And so I started with the genomes from Green et al. This is basically a tetrapod tree. This is a UCE tree, so it has about 4,000 loci in it. I simulated read data from these 4,000, 5,000 UCEs across the tips. So I basically did this read simulation for each of the tips in the tree. I only used ART this time. I reassembled the data for all taxa with Trinity, so I didn't do all 3 assemblers this time, mostly because I ran out of time. And I aligned those with math, clipped the alignments to the same length. I'm skipping all the details on that. If you have questions, just ask and I'll tell you. And again, I wanted to perform a sanity check. So the first thing I did was simulate reads without incorporating the sequencing error. And the nice thing is that the topologies, as you would expect, are identical between the data that I started with from Green et al. And the data where I've not added any sequencing error. This is good. It should make you feel a little bit better. Once I added assembly error, and when I looked at the data with 60% reads on target, I also get exactly the same topology as what I started with. This should also make you feel relatively good. The topologies are the same from what we started with and the simulated data. As I reduce the number of reads on target to 20%, I also get the same topology. So topologically, we're getting the same result. When we look at branch lengths, what you would expect if there's not a problem is that there's a one-to-one correlation between the branch length and the data with no error and the branch length where we've added error to the sequencing reads, and that's in fact what we see, right? So again, you should feel pretty good. So sort of the remaining question is, are you still screwed by these problems? And the answer is I don't entirely know yet. You might be wondering too, what's going on? Why is it that this isn't having an effect? And one of the reasons is that the sequencing errors that we get are roughly random. They're semi-random, and we can talk about the distribution of those, but let's just call them semi-random. And what we ended up with is assembly error or additional noise in a sea of informative sites. And those sites were basically, the signal was the overcoming noise that was introduced. Small errors may still lead to big problems, and that may affect gene tree inference, where we have very few sites that are making a big difference. And the other thing is, there's some preparation steps that go slower or will take longer because we have reads with error. The last slide I have is just showing that we can actually treat these sequencing errors by doing raw read error correction. And once we do that, we basically see that relative to what we started with, things like Abyss and Velvet perform better when we're correcting the sequence errors in the data. And with that, I'm